Hello students, welcome back to Agriculture Form 3. We are still on the mushroom production. This topic is a little bit long. Last time we discussed on the two lessons whereby we described the mushroom fruit. More especially we looked on the parts and their functions. Then later on we looked on the importances of these mushrooms. Then later on, we started discussing on how best we can manage the mushroom. So we looked on the factors to consider when you are selecting mushroom species. We also looked on the factors to consider when you are selecting the site where we are going to construct the mushroom house. As we learned that these mushrooms, they are cultivated in a house. So we looked on the conditions of the house where we're going to grow our mushroom. And now let us proceed from where we stopped. Be with me as we go through this topic. So on our today's lesson, we are going to concentrate much on substrates. On this lesson, we are going to concentrate much on substrates. So we are going to define the term substrates. Then later on, we are going to list examples of substrates used in mushroom production. And lastly, we are going to describe how these substrates are prepared. How the substrates are prepared. So, what are substrates? A substrate is a special material which is organic in nature, prepared for the purpose of planting mushrooms. So initially, these mushrooms, they are not planted on the bare ground, but rather we plant them on organic materials. So we have special organic materials where we plant the mushroom. So these special materials which are organic in nature and which are prepared for the purpose of planting mushrooms, we call them substrates. We call them substrates. So what are these special materials which are organic in nature, which we prepare them where we can grow our mushroom? So materials used as substrates may include rice straw, maize stalks or sheath, sawdust, molasses, fertilizers. Sometimes we add some inorganic materials so as to boost production. Instead of fertilizers, we can just add chicken manure. We can also use soya and bean powder. So, how are these substrates prepared? These substrates, they need to be prepared very well in the sense that these mushrooms are very delicate. The mushrooms are very delicate. If the substrates we are going to prepare, they're not well prepared, sometimes that might reduce production or even those mushrooms might not even grow very well. So we prepare these substrates in two phases. So substrate preparation involves two phases of composting. We have phase 1 and phase 2. In phase 1, this will allow the multiplication of microbes, hence increasing microbial activities and soften the substrates. So when we are preparing the substrates in phase 1, we are just trying our best to multiply microbes. These microbes will help to partially decompose these materials. Where we are going to plant our mushroom, we don't want the materials which are very tough or which are very hard. At least we need to soften them. So these mushrooms are softened, these substrates are softened in phase 1. So we're saying in phase 1 allows the multiplication of microbes, and these microbes will help to partially decompose these substrates. So this will be through increasing microbial activities. So how we do that, we are going to explain into details. Then we have, we prepare these substrates and the phase 2. So phase 2 help to eliminate germs or microorganisms which may destroy mushroom through pasteurization or sterilization. So these substrates, remember, in phase 1, we are saying that we need microorganisms. Microbial activities are needed in phase 1 just to make sure that the substrates, they are partially decomposed. But in phase 2, we are trying to eliminate the microorganisms. In phase 1, microorganisms are needed for partial decomposition, but in phase 2, we no longer need these microorganisms. Before we plant our mushroom, we eliminate all microorganisms, including the germs. So, just because these microorganisms or these germs may destroy our mushroom. So, to run away from our mushroom being destroyed, we eliminate the germs, any germ or microorganisms present in the substrate. So this is done through pasteurization or sterilization. So we are also going to learn into details how pasteurization as well as sterilization is done. So be with me as we go through substrate preparation. So under phase 1, uh, that is substrate preparation. This is also called outdoor composting. Phase 1 substrate preparation is also called outdoor composting since the procedures are done outside the house. Of course, they are done under the shade but should be outside the main house. So under this phase, it will just involve wetting and mixing of the composting ingredients. Remember, we have listed several ingredients or examples of substrates which we use where we can grow our mushroom. So those substrates, they are just they are mixed all together. So we mix one substrate after another. As we are mixing those substrates, we also apply some water. So we apply some water, then we mix, and this whole process will take place within 16 days. So this whole process will take within 16 days. So mark this under phase 1 substrate preparation. It much involve wetting and mixing of compost ingredients. So I'm saying that these ingredients are mixed one by one. As we are mixing, we apply some water. Why are we applying some water under this stage? Remember, phase 1 will make sure that the microorganisms should be available. We need microbial activities. So as we are applying the water, that will provide a conducive environment for multiplication of microorganisms. So wetting of the compost with water will help to multiply microorganisms, which will help in partial decomposition. So I'm saying that this process under phase 1 will take place for 16 days. After we are done outdoor composting, that is in phase 1, these will go through phase 2 now. The substrates will go through phase 2. So under phase 2, this composting takes place inside the mushroom house to pasteurize and condition the compost. Remember, under phase 2, we are trying our best to eliminate microorganisms as well as the germs. So to eliminate these germs or microorganisms, the substrate should undergo pasteurization. Pasteurization is accomplished by introducing live steam generated from a boiler outside the house through metal pipe into the mushroom house. So what happens is that inside the mushroom house, inside the house, you are going to put your substrates, the partially decomposed substrates. Then outside the house, you are going to have what we call a boiler. That boiler, it's like you'll be boiling the water in that boiler. Then you connect the metal pipe from the boiler, connecting it into the house. So the hot steam will be coming out from the boiler via the metal pipe. So that live steam, the steam from the boiler, which is too hot, will try to kill the germs as well as the microorganisms from the substrates. So this phase lasts for 12 to 14 days. It lasts for 12 to 14 days. Should be a continuation of phase 2. So the higher the nutrient content the substrate has, the higher the mushroom yield. That much we should know. Let us now look into details what transpires in phase 1 and what transpires in phase 2. Here is the video showing mushroom substrate preparation using the boiler. Remember, we're saying that once the substrates have been partially decomposed by microorganisms or microbes, and those microorganisms need to be eliminated. So we can use the boiler, which will be introducing the hot steam into the house. So let's take a look on how these boilers work.
This is a baby boiler for mushroom cultivation. In this pipe, the steam flows, and this is the oven on which the boiler sits on. Here we put timbers for burning fires, using which we actually boil the water inside the drum, which is the actual boiler. In this machine, separately a water observer, a safety valve, and a chimney from the mid side, and this portion is for water inlet whenever water goes down from a certain point below, and then we switch on our motor and fill it up again, so it can produce continuously steam, and we can just put it on inside any vessel for collecting the steam and sterilizing the substrate.
Yeah, so that's it. As you can see, we have the boiler, and up there was a metal pipe. That metal pipe is connected from the boiler to the mushroom house where the substrates are. So if there is heat down the boiler, then inside should be water. So when the water is boiling, it will produce the steam. So that hot steam is the one which will go via the metal pipe into the substrate, the house where the substrates are kept. So through from the hot steam, that hot steam will be killing the microorganisms as well as the germs. Now let us look on the details how these substrates are prepared in phase 1. So we have a table here showing the day as well as the activities which are done on each day. So on day 1, what we do is pre-wetting of straw. So most of the times we start with the materials which are very hard. So on day one, you just do the pre-wetting of hard materials. Like in this case, if you are to use the straw, it can be the rice straw. Then day 2 to day 4, you add water to straw if necessary. If on day 1 you added more water, it means on this day day 2 to day 4 you will not add more water. But those straw, if they are dry, day 2 up to day 4 is just a matter of adding water. Then you come to day 5. On day 5, it will be the first turning of compost. So you turn the compost. In this case, we are talking of the straw. After you have added the water from day 1 up to day 4, you just add the water. Then day 5, you do the training. The upper part should be lower part now. So first turning of compost. As you are turning the compost, you also add other materials. That's on day 5. So you add white lime, you add chicken manure, and half amount of sulfate of ammonia. Then day 6 and 7, these days you just leave the substrates like that. You just leave the substrates like that. Then you come on day 8. Day 8 is the second turning of compost. So you turn again that mixture. You turn the mixture. On day 8, by that time it means you have a mixture of straw, white lime, chicken manure, and half amount of sulfate of ammonia. So after turning on day 8 the compost, as you are turning, you also add other materials. On this day, you add uh, uh, static lime as well as this single super phosphate. Then on day 9, you leave the mixture like that. You don't do there's no any activity on day 9. Then comes day 10. On day 10, you mix and wet soybean powder or cotton seed cake, rice bran, and molasses in a drum. So you put these substrates separate on a drum. So you are going to mix wet soybean powder, cotton, or instead of soybean powder, you can use cotton seed cake. Then you add them to rice bran and molasses. So you put the three mixture in a drum, separate in a drum. This you not add with the mixture which you have composed in day 8. So this is a different mixture. You do it separate in a drum. Then on day 12, day 11, it means you are just going to leave the mixture. In this case, you have two mixture. The mixture which you have prepared on day 10 and the mixture which you have prepared up to day 8. So on day 11, you leave the two mixture. You just leave the two mixture. Then on day 12, there is third turning of compost. So that compost which you prepared up to day 8, you turn that compost. Then as you are turning, you add the wetted mixture which you prepared on day 10 and the remaining sulfate of ammonia. So on day 12, you do the third turning of the compost prepared up to day 8. Then you also add the mixture which was prepared on day 10. Then day 13 and 14, you leave the mixture like that. Day 15, that's the fourth turning, whereby you loosen the compost. Then day 16, you do filling or transferring of the compost onto growing beds up to 10 centimeters deep. then you close all ventilators and doors. So that's the whole process, what transpires on phase 1 substrate preparation. So all in all, in short, we are saying that on phase 1, it's just a matter of pre-wetting and training the compost. It's just a matter of adding the water, then turning the compost, the mixture of the compost. Let's look only phase 2, the details, what transpires in phase 2. Remember, on day 16, more especially when we are looking on phase 1, we said that we put those substrates in a house. So after you have put the substrates in the house, day 17 starts phase 2 of substrate preparation. So on day 17, there is introduction of live steam into the mushroom house. And day 18, you maintain compost temperature at 65 to 70 degrees Celsius, at least for 4 to 10 hours. Then day 19 to 21, you try to decrease the temperature slowly. So from 65 to 70 degrees Celsius, you decrease it to 50 to 52 degrees Celsius. How do we decrease the temperature? We do not reduce the amount of heat, but rather we just open the vents. We reduce the fire and open the vents. Then on day 22 to 25, you further decrease the temperature to between 40 to 45 degrees Celsius. So if you just open the windows on day 19 to 21, then on 22 to 25, day 22 to 25, you further open other windows. Then day 26 to 28, you aerate and cool compost to 25 degrees Celsius by opening all the ventilators up until the substrates are cool. Then day 25 to 30, if there is ammonia odor and the compost has high moisture content, you continue aerating up until all the odor is no longer there, as well as the temperature is not that high. It means reaching this far, the substrates are well prepared. They are well prepared, the substrates. So when we are treating the substrates, more especially under phase 2, there are several methods which we can use. After we have prepared the substrates, it should be it should be treated first before mushroom are seeded, before seeding the mushroom. So there are methods used to treat substrates in mushroom production. When we are discussing into details under phase 2 how to treat these substrates, we've mentioned only one, that's steam pasteurization. But there are other methods which we can use apart from steam pasteurization. So we have immersion in hot water. The substrates are immersed in hot water. We can use chemical sterilization technique, or we can use what we call sterile technique. Let's go through on these methods of treating substrates in mushroom production. So for steam pasteurization, I think we have discussed much. Substrates are packed in wooden trays or boxes and then kept in a pasteurization room at 60 to 80 degrees Celsius for a few hours. And the temperature of pasteurization room is manipulated with the help of steam through a boiler. So that's what we were explaining when we were discussing in phase 2. We said that the steam is introduced into the house. So the substrates are packed in the wooden or boxes and they are left in a house. Then you introduce the steam. The steam will be coming from a boiler. So of course, I mentioned that you connect a a metal pipe from a boiler to the house. So the steam will be coming from a boiler via the metal pipe into the house. Then the substrates will be pasteurized. After cooling the substance to room temperature, then it is seeded with spawn. So once the substrates are well treated through pasteurization using the steam pasteurization, then later on we are sure that most of the germs are eliminated or most of the microorganisms are eliminated. Then those substrates are ready for seeding. So the entire process can take 3 to 4 days. It will depend on the uh, amount of heat which is produced. Apart from that, we can use what we call immersion in hot water. The substrate is soaked in hot water. The hot water can range between 65 to 70 degrees Celsius. So after they have been choked between 5 to 10 centimeters, you can put them in hot water. So they can last for 1 hour or 30 to 60 minutes. That is they can last for 1 hour if we are using the temperature, the water which is of temperature between 65 to 70 degrees Celsius. But if the temperature is 80 degrees Celsius, you leave the substance between 30 to 60 minutes. But in case we have padded straw, which are very hard, make sure that the temperature of the water should be up to 85 degrees Celsius. So those straw can just be there, can be soaked in 85 degrees Celsius water for 30 to 45 minutes. Then later on, the water is drained. So after draining excess water, then spawn is added. But make sure that the substance should be not hot, should not be hot, should be cool. Apart from, hot water will help to soften hard substrates, further soften hard substrate. Remember, on phase 1, we do partially decompose them, but still more, some of the substrates will still be hard. So if they are still rough, hot water will soften them and hence easy growth of mycelium. But apart from that, this hot water, the other use of hot water is to get rid of microorganisms and germs. And this method is not suitable for large-scale mushroom production, just because we just soak the hot water will be in drums. So the drums may not accommodate more substrates. So if you have a large scale mushroom production, it means you need more substrates. And these drums, they may not accommodate many substrates. So that's why we're saying this method that is immersion in hot water is not suitable for large scale mushroom production, but rather for small scale mushroom production. Apart from that, apart from the two methods, we can also use chemical sterilization technique. So this is done to kill competitors, molds, or suppress them. So it's just a matter of using the chemicals. So the substrate, you put the substrate in the water with some chemicals. So those chemicals will be the ones which will eliminate the germs as well as microorganisms which might disturb the growth of mushroom. Or sometimes we can use what we call sterile technique. Under sterile technique, first of all, the chopped substrates are soaked in cold water, very cold water. Why in very cold water? The very cold water, most of the times the microorganisms will not be active. The microorganisms will not be active, so they will not multiply much. So the soaked substrates are then put in heat-resistant polypropylene bags and sterilized in an autoclave for 1 to 2 hours. So once you have soaked them in hot water, then you remove them. You remove those substrates and dry them. Then later on, you pack them in a polypropylene bags, and you put those substrates while they are in the bags, you put them in an autoclave. So this on the picture, it is showing an example of autoclave. So these are the bags we are talking about. The substrates are packed in these bags. These bags are resistant to heat. So you pack them in these bags and put them. This is the autoclave. It's the same like this one, only that there is an opening here where you are going to put the packed substrates. So after 1 to 2 hours, you remove them, you let them cool down, then spawning is done. So that makes the end of our lesson. And on this lesson, we focused much on substrate preparation. So far, we have defined what substrates are. Then we have also looked on how best we can prepare these substrates. Remember, we've emphasized that these substrates, they are prepared in 2 phases. Phase 1, which is also known as outdoor composting, then phase 2 of preparing the substrates, whereby this will just involve pasteurization or sterilization of substrates to get rid of germs as well as microorganisms. So when we are getting rid of germs or microorganisms, remember there are 4 methods, and we have described all the 4 methods. So if you have any other questions, you can use the WhatsApp number or just visit Xulendo line so that you go through again this lesson for you to fully understand. And these are the resources which we have used to come up with this lesson. You are also free to go through these books as well as even the pictures or the videos from the internet so that you fully understand the topic. So we are going to stop here. You were with me, Dorothy Maowa, your usual agriculture teacher Form 3. Until we meet next lesson, goodbye.